I'm going to draw a thing. So this is what it's going to look like. This is like your, I would put my paper in landscape mode if I were you. Because I'm going to draw. So these are the edges of your paper. This is the top of your paper. This is the side of your paper. That's the bottom of your paper. And then this actually, I want this little crossbar to be a little bit low, not quite half.
How large should this be?
This is your whole little sheet of paper.
Oh, wow.
For a reference. So that's what it's going to look like. And really, if you have to air two of these boxes, technically the top two should be bigger than the bottom two, but we can squeeze it all in however you do it. Okay, I cannot get my stupid video to video the right thing. It's really bothering me. Video, you're bothering me. Just FYI. Okay. So biochemistry is unit 1. It's basically water and the 4 major macromolecules, okay, and then kind of how they work. Some of this we'll pick up even greater detail when we do their review over like DNA replication. Then we'll talk about the 5', 3', and all that stuff. So we're not going to go into that today. We're basically just getting an overview of our macromolecules. I have this little circle in the center. So each of the macromolecules is going to have their own little quadrant here. But I have this little circle in the center because there's 2 things that go with all of them that affect all of them, that kind of span the whole unit. So we're going to put them in the middle. And the first one is water. Okay, so water. I draw my little Mickey Mouse guy. You got your hydrogen and you got your oxygen like that, okay? And yeah, he's a wee small, so hopefully you can see him. But the little ears are hydrogens and the big thing is oxygen. So just in case you don't remember, this is what we're doing is we're going over things in case you don't remember, so I don't have to say that every time. So they have a certain type of bond between them. They have what we call a covalent bond, okay? Covalent. Co means both, and I'm gonna spend a lot of time in our review just reminding you what root words mean and all that stuff, because that is super helpful in biology. Like when you're trying to remember what terms are what, if you can take that word apart, so much better than trying to memorize flashcards. Okay, so covalent, co means both. And then valence, well, that's referring to your valence electrons, like your valence shell, okay? So covalence means it's like they both have the same valence shell, like they're sharing their electrons. They're both, the electrons belong to both of them, okay? Like co-parenting maybe, right? So the kids belong to both of them. And this is actually a pretty good example, because just like some of our co-parenting relationships and things, they're not exactly equal. Like one parent has the kids more than the other, right? So oxygen actually has the electrons that are shared more than the hydrogen does, which creates a situation where oxygen is a little bit negative because the electrons are negative, while the hydrogens are a little bit positive, okay? So in the end, this is a polar covalent situation because it has poles. It has a positive pole and a negative pole. We've got a charge, okay? And this sets up water for its properties, which is the important part. How does water behave? Like what does it do for us in life? Because that's what we care about. This is biology. So basically, since you have a negative side and a positive side, you're going to have attractants, right? So the negative is going to attract positives. Well, it can attract other water molecules, right? And so then we would call that, here's co again, cohesion. Okay, adhesive we know means like sticky, like it's sticking together, and co is both, so they're both water molecules, right? So they both of them are water and they're sticking together. Okay, so that would be like if my hand is a little water molecule and my fingers are the hydrogen side and my thumb is the oxygen side, it's like they can, they're gonna, opposites are gonna attract, and so then you'll get them where they'll all start forming a sheet, okay? And that's important. One thing that cohesion does, and so these are both water molecules, so water sticking to itself, it creates something called surface tension. which is, it's important in biology in a lot of different ways, but cohesion itself is important in biology in a lot of different ways. But surface tension, they talked about actually in one of our multiple choice questions that asked what was happening here. Why did it sink? And it sunk because it broke the surface tension. It was a surfactant. So that's one thing they expect you to know. The other thing is with a charge like this, it sticks to other things. So like it sticks to the negative side will be attracted to things that have a positive charge, and the positive side will be attracted to things that have a negative charge. And so water sticks to stuff. You know this because you experienced water. You know that when it rains on your car, the water droplets actually stay on the window and roll down rather than bouncing off. If you've ever used like a Rain-X or like some sort of windshield, it's like a wax. I don't know if you've ever heard of this Rain-X business, but basically there's Rain-X and you just squeeze it on a rag and wipe it on your windshield, and it's see-through, but it's like a waxy coating. If you have that, the rain actually bounces off your windshield, and so it doesn't stick, and so you don't actually need to use your windshield wipers. So that's kind of weird. But it's because it's a lipid, because it's a wax, right? So it can't stick to the lipid. But otherwise, we know that water does stick and rolls down rather than bouncing off, okay? So water, let's see, what else did I need to know? I have all the standards here. Can you grab that door? It's one of my freshmen who wants to retake a quiz, I'm pretty sure. So I have the standards here in front of me to make sure I don't skip anything. Need to retake, or you wanted to see? I'll just open it for you if you want to go find a spot. Yeah. Okay, so let's see. Water. Got that. Cohesion, adhesion, surface tension. Any questions?
What was the difference in cohesion and adhesion?
So cohesion, it's here. We just think of they're both water. And adhesion is to other things, like adhesive, like tape sticks to, stuff. So this would be like water sticking to other things besides water, and cohesion is water sticking to water.
Gotcha.
Okay. All right. So next up, we're going to take each of our 4 macromolecules. Oh, just kidding. Next up, we're going to tell you how we put them together first. Let me get Aaliyah Grace settled with her. Good. Okay. So, do you remember what a monomer is? It's like a single little piece of something. So, what is the monomer for a protein? Anyone remember? Amino acid. That's correct. Amino acid is the monomer for protein. What is the monomer for a nucleic acid? Anyone remember? What makes up a DNA? What's the little piece called? Nucleotide, right? Nucleotide. Okay, we got to memorize these, so make sure you have them memorized. Okay, Aaliyah Grace, you should be able to open that. Let me know if you can't. I'll put them on here. Libby, I'll put them on here. Yeah, I was just reviewing what a monomer was, actually. So if you have a monomer, and you have lots of monomers connected together that make a polymer, right? Does everyone remember those 2 terms? So a monomer would be like a single repeating unit, like amino acid, amino acid, amino acid, amino acid all stuck together, right? And then they would make a polymer, the protein. Yeah? Everyone chill with that? Okay, so we need to be able to know the terms from going from a monomer to a polymer, or from breaking up a polymer and going back to monomers. So this is something that's kind of a big deal. Okay, so let's see if we can get this, because we've had it a couple of times on a couple of tests and we've missed it every single time. So let's quit missing it immediately. All right. So I'm going to draw a little line. So hopefully that makes sense, going from a monomer to a polymer, right? Like building it. So let's get the vocab down. So if I'm doing this, am I synthesizing something or am I lysing something? I'm synthesizing. Okay, so that's the first part, is you are doing synthesis. So the term is actually dehydration. Synthesis. Okay. Huh? This has been on our test several times. Unit 5, about 3 weeks ago, we took the Unit 5 test. Multiple choice question. Most missed question on the test was simply dehydration, synthesis, and hydrolysis is all it was. Okay, it was just you had to break it apart. and look at the pieces. So I'm going to try to get that question pulled up so that you can see it eventually. Okay, I'm going to put the words on here and then I'm going to explain them. Make sure you know them. Okay, on the flip side, if you're going from a polymer, if you have a long chain of amino acids that you're breaking up into, and you're going to break them up into just their amino acid parts, right, instead of a long protein. Yeah. Oh, yeah, okay, I need to take that longer for you. Okay, so then you're going from polymer, you have a big protein, and you're going to break it into amino acids, you're going to break it into its monomer, then that is basically called hydrolysis or hydrolysis. Okay, we actually call it hydrolysis, but if you break the word apart, it's hydro, water, lysis, breaking apart. Okay, yes.
So monomers are always connected by water.
Okay, so let me explain it. Just 1 second. Sorry, I'm got to fix a setting for her. 1 second. Okay, I fixed your setting. Okay, so what happens is you have like little pieces, let's say amino acids, right? So you're doing translation, ribosomes spitting out, pulling in the amino acids and putting together protein. Okay, so on each amino acid, there's an end that has a hydroxide, an OH, right? It has a hydroxide on one side. On the next amino acid coming to join the train, there's a hydrogen. When they get close enough together, that hydroxide will bond to that hydrogen, creating H2O. That H2O leaves. See ya. Leaving a little, like, if you want to think of it like trailing ends that can bond together, the two amino acids, okay? So it's dehydrating and synthesizing at the same time, right? The water is bye-bye, and the synthesis is happening that way, okay? So removal of water going from monomer to polymer. Okay, now on this side, same thing. So now you have the long chain protein, little amino acids all in a row, like a little train. Now you have them because this is the polymer, right? But it's going to be broken apart. Now this is going to require a little energy. A little ATP is going to have to help us out. But what's going to happen is when the amino acids are all in a little chain, the little chain is going to get broken, right? Because that's going to require a little bit of energy to do that. And when it does that, then a water molecule can once again be pieced into a hydroxide on one side, hydrogen on the other, and they go away and are like done. And they're broken, lysis, by a water molecule. Okay, so water breaking, and that's what hydrolysis means. Now, if you don't, I don't recommend like the whole flashcards thing like that. That drives me nuts, but maybe you're a good memorizer like vocab like that, and that's fine too. I prefer to remember what the words mean. So dehydration synthesis, like synthesis is basically just synthesizing something, making something, and it's doing it by pulling water out of the system, okay? And this one, lysis, that is a big word right there. Lysis for biology, breaking. So if a cell lyses, lysosomes, hydrolysis, all this lysis, that means breaking, and it's using water to break it, okay? So that's going from polymer to monomer.
This hydrolysis doesn't remove the water though, because it just
it actually adds the water in.
Okay. Oh, wait. So then, what is dehydration?
Dehydration is removing. Like, think about it. If you're getting dehydrated, the water's leaving. Yeah. Okay. Mm-hmm.
So dehydration synthesis would be exothermic, and then hydrolysis would be endothermic.
I hate to say yes as a rule, but I think so. Just because I'm just sitting here going, pull energy in? Yes. I'm just not 100% confident in my yes. But that's my understanding. Okay. All right. But it doesn't have to be written on your stuff because it's not in your standards. Okay. So, but this is absolutely, you have to know that. Like, you guys have missed questions throughout the year because we cannot remember that. And it's like we go over it and it's like we just forget it. So anytime something's being built, in biology right now, because there are other ways, but in AP biology, freshman level college biology, if it's being built, it's dehydration synthesis. And that includes any of our 4 macromolecules. Okay, if it's breaking apart, it's hydrolysis. Okay, and there, like I said, there's other ways, but those are our 2 main ones. Okay, all right. So then let's look at what we're building with this. So I've got 2. I'm going to put at the top here. So we're going to start with, I don't know how your paper's set up, if you have a spot over here or if it's just going to be under it, but wherever. These are nucleic acids.
Is this the bigger sign?
The top, yeah, the big ones. These two are the big ones. So nucleic acids and proteins. I'm going to de-squinch mine a little bit here. Okay, so nucleic acids, proteins, and then down here it's going to be carbohydrates. And it's going to be lipids. Okay, if you have to spend just a little bit of time because you're out of time, you want to review, and you want to review Unit 1. A lot of you guys that actually were able to complete your mock analysis told me that macromolecules were low on your list of confidence. And that's just because of the nature of them. That's very common. The deal is if you want to take the time to review Unit 1, to review your macromolecules, spend time with nucleic acids and proteins. Don't worry too much about carbohydrates and lipids, okay? There's a couple of things you need to know about carbohydrates and lipids, but they're not that big of a deal. Nucleic acids is DNA, so just a wee bit important. Proteins are freaking all over the place. Every single unit deals with proteins. It feels like the test is almost a protein test because it's all about proteins and enzymes and activation energy and denaturing and active sites and all this stuff, right? But it's because proteins are literally that important to biology. And so that's where I'd spend my time. I'd spend my time on these two. If I had to choose between the two, I'd spend my time right here, okay? Because that's going to get you the most points for your review money, basically, okay? But let's start with nucleic acids really quick. So this is our example here is DNA and RNA. And by the way, DNA, we tend to just think, oh, there's just one type. And there is, but we are going to look at the difference between prokaryotic and eukaryotic really quick before we're finished today. But DNA is pretty much DNA. It is a highly, highly conserved molecule, right? All organisms are very, very similar as far as DNA goes. So that's kind of a big deal. So it is made out of carbon, hydrogen, oxygen, and nitrogen, and phosphorus. Those are its elements. So it's CHOMP. Okay. And its function is basically inheritable genetic instructions. Okay, and we know, because we're awesome biology students, that its function really is to create proteins. That's what it does. I mean, it has the code to create the proteins, but it is inheritable. You do pass it on. You don't inherit proteins. You inherit DNA, and then DNA codes for proteins, right? So this is our inheritable portion. Okay, its monomer, so then structure here. So its monomer is a nucleotide, and I'm going to draw a little one here, and then I'm going to go over there and enlarge it so that you don't have to try to read my tiny writing. Okay, I drew a purine, right? Because it has 2 rings, and then pyrimidine would only have 1, but that was just for fun. Okay, all right. So over here, let me switch back, make sure I can see my video here. Okay, over here, I'm going to draw on a little, I'm going to enlarge it, but you can just label yours there. It's just that gets so small that you can't see it. So I don't want you to not be able to see what I was drawing. So here's my little terribly drawn purine, looks like some sort of bad dog. Coming out of the dog house, he's chained up. Okay, that's bad. We shouldn't chain up our dogs. Okay, so this is your phosphate. And that is PO4 negative, if you must know. Oh gosh, I'm stepping on things. And this is your sugar. And this is your nitrogenous base. Right there. Okay. Put my little recording over there so I can see it better. Okay, so that is the monomer that makes up DNA. Okay, that makes up nucleic acids. It doesn't have to be DNA, it could be RNA. Okay, so that is your nucleotide. So you can label it in your little spot over here if you want to. All right. Okay, so now that we've got all that done, we have 2 different things we need to talk about. One of them is DNA versus RNA. make a little chart like that. I have probably allergies, but you never know around here.
Wait, both of those are the nitrogenous.
Nucleic acids made of nitrogenous bases. Wait, oh, both those little things? It's a double ring.
Okay.
Purines, 2 rings, and pyrimidines, just 1.
Why is there what's the difference in their function?
Purines and pyrimidines, like guanine and adenine are purines. Oh God, now I have to think. Yes, purines and thiamine and cytosine are guanine. Good Lord, pyrimidines. Yes.
What's the word after genetic?
Instruction.
Okay, sorry.
It's okay. And on top of that, I'm coughing, so that's annoying. Okay, so DNA, you have A pairs with T, and I'm going to double my stuff here, and G pairs with C. Do you notice how many? Can you see how many bonds are between them?
2.
2 for,
A and T.
yeah, and,
3.
okay, you can see that. Okay, 3 for G. So, technically, A and T is a weaker bond than G and C. They're hydrogen bonds, both of them are hydrogen bonds. It's just you have 2 for A and T. So when we're doing our PCR and we're heating it up to denature it, make it fall apart, right? So that we can actually get in there and make copies of it or whatever. When we do that, the adenine and thymine fall apart first. And the guanine and cytosine hold longer because they have a 3rd bond. Makes sense, okay? And over here, I'm not going to bond anything together because RNA doesn't technically, literally bond. It's single strand, right? So it doesn't bond. It does use that base pairing rule to pair up with DNA, but they don't hydrogen bond together like they're one molecule. So I'm taking the bonds out of them, okay? So this is a double strand, and this is single, and this has deoxyribose. Whoops, that is a bit. And this has ribose for its sugar. Okay, they're both a pentose. Both of them are pentose sugar.
What's the difference between deoxyribose?
It's just a slight chemical difference. Both of them are technically C5 carbons, so C5H10O5, but they have just slightly different bonding inside of them.
Okay. Do you remember that? I don't know if you remember. There was a question on the test. That was about DNA and it had the chart and everything. But when you fold it, there was not an option.
It may have been a double-stranded RNA. Was that an option?
No, you had to say what it was, LEU or.
Oh, I don't know. I'd have to look at it again.
Oh,
I don't remember.
So
Okay.
No, it was
the Unit 6 test we just took?
Yes.
Okay, I'll look at it.
Deletion. It was what caused this mutation or something.
Okay, I'll look at it again. I haven't looked at it close enough to know. I just got them during lunch.
But I also have my lack of knowledge on that. So.
Okay. Sorry. Okay, so finally, you need to have another little thing that talks about the DNA for eukaryotes versus prokaryotes. And this barely fits, but okay, hopefully you have more space. Or you could do it down here if it fits better. Okay, so put it wherever it fits best. There's just 2 words. I'm going to write 2 words on each side, so it's not doesn't have to have a lot. Okay, but there is something important we need to deal with here. Eukaryotes have linear DNA, means it's in a line. Okay, typical chromosome. What do you think? Chromosome 1, chromosome 2, chromosome 3. There's a beginning and an end to each of those chromosomes, right? Okay, and then also it is in pairs. 1 from mom, 1 from dad, right? So you have 2 linear chromosome 1s and 2 linear chromosome 2s. Not so for our prokaryotes, which have circular, and that just didn't work. Circular, and they are single. Okay? All right, see if I can get this across to you so that you never ever forget it. We're probably gonna run out of time, shoot. Mitochondrial DNA. Number 1, missed on our free response for our mock. Mitochondrial DNA and chloroplast DNA, mtDNA and cpDNA. Mitochondrial, chloroplast DNA are right here. They are prokaryotic in nature, which means when the question says, why is this trait rarely ever heterozygous? Because there's only one, okay? You can't be 2 different One big A, little a, if you only have one A, right? So that's why mitochondrial DNA is not heterozygous. It can't be. There's only one. So you can't have big and little when you only have one. You're either dominant or recessive for mitochondrial traits. Just the way it is, okay? So, cool. Questions on that? So you might want to make note to yourself, like up here somewhere, maybe that's mtDNA and cpDNA. Oh, DAN, Dan? I don't know what I'm writing. They go with prokaryotes, okay? They just like prokaryotic DNA, which is why endosymbiotic theory is a thing. Okay, let's do proteins really quick, and then I'll slap on our, I can do proteins in about 4 minutes, then I'll slap on carbohydrates and lipids, and we'll call it good. Okay, so for this one, our example, I'm just going to use enzymes as an example. I want you to remember that enzymes are a protein. They're a class of proteins. So if you know how enzymes behave as far as denaturing and shape and active site and all that, then you know how proteins work, okay? So these are actually formed from CHONS, sulfur, instead of phosphorus, okay, which is the only difference. I mean, that's how you can kind of tell if it's DNA or protein. If you have a mess in your hands and you're trying to determine which molecule it is, you can look for sulfur or phosphorus, okay? Its function is basically all cell processes. If you want to make a list, you could do like motor, like movement, transport. You have like protein channels, you've got support, you've got enzymes which lower energy activation, like you name it, proteins have a hand in it, okay? So I typically don't even write them out. Monomer for proteins, we already went over it. Amino acids, I'm going to draw one. Okay, we're a carbon base. Then we have a carboxyl group. Okay, you really have to recognize this, not necessarily draw it, although I am getting better at it in my old age. Okay, so here's one. Actually, I guess I don't need to enclose the bond. Hang on. Let me box in the right thing. There we go. 1, 2, 3 boxes. Okay, those 3 boxes you need to be able to recognize, or at least not freak out if they use the terms. Some are more important than others. Okay, so this is an amino acid. So that's what this is. It's amino acid. Okay. We're going to lock them together like a train is what we do with amino acids. Okay. So this is carboxyl. And this is the amino group. So this one's, that's an A. Amino group, carboxyl group, and this one is the R group. What a nice name. Okay. Cool. The R group can be different for each amino acid, right? This is the only part, really. The amino group, the carboxyl group, they're just kind of the same. And it's easy to remember which one's which because carboxyl is literally carbon oxygen. So it's like, it just says carboxyl, and that's exactly what it is. If it has a nitrogen, it's not carboxyl. This has nothing to do with carboxyl. It doesn't really have anything to do with amino either in my brain, but at least I know it's not carboxyl. And that one, if I can't figure out that's the R group, there's something wrong with me. So, okay, so that's just it. You got other problems. Okay, so basically we are going to have 4 levels of structure, and this is the important part for proteins. And since form equals function, it's pretty important we have our form right. So this primary structure is simply the order of amino acids. I'm going to put AA for amino acids. Secondary is bonding between amino acids, not R groups. And then tertiary is the folding based on R properties. Okay, I remember those are going to be things like hydrophobic, hydrophilic, and our stupid standards call acid and base ionic. It's annoying. Okay, so primary is just your order of amino acids. Secondary is the bonding between the amino acids. So it'd be like bonding between, a lot of times it's this hydrogen up here is actually what's bonding between those. So things like, that's when they coil up and make an alpha helix, or they make a little pleated sheet or something. But primary and secondary have nothing to do with the R group. This is the one that actually folds it into the shape where its form and function is kind of a thing, right? That tertiary structure. If the tertiary structure is off, you got no function, okay? Your active site doesn't work anymore. And then quaternary is just the joining, I'm not going to say bonding, joining of subunits. And so that would be like, basically, if you've got 3 or 4, like an alpha, beta, gamma usually is what it is, alpha, beta, gamma, little things, like here's a little hemoglobin alpha, hemoglobin beta, and then they all make a big one. That has quaternary. Most of our proteins don't have a quaternary structure, okay? All right, let's do these really quick. Both of these are CHO. Okay, and so really all you need to know of these is on carbohydrates. Example, let's just slap glucose on there because it's the most common one. If you don't know the formula for glucose, there it is, C6H12O6. Function, short-term energy, and structural. Okay, they can often be linear. I'm not going to put any hydrogen on there because my brain will blow up and I don't want it to. Or they can be like, you know, like a ring, like little carbon sticking off the ring. A lot of times it's based on where they're at, but mostly they're linear. Okay. Carbohydrates. Really, that's all you have to know about them. Structural, think cell. Here, let me put in parentheses, cell wall. Like cellulose, you know, cellulose is cell walls. Oats, sugar, carbohydrates, okay? So like cell walls is where you'll find those. And then finally, so really that's what I'm saying. These two are your big ones, right? They have tons of information. You got to know the nitty gritty, all the little names and stuff. And this is like, just know that, you're good. Lipids, our favorite example for a lipid in AP biology is a phospholipid. Absolutely best one. What you need to know about these is they're hydrophobic. Look, I'm making a little funny thing. Hydrophobic. They hate water. all the other macromolecules are like attracted to water. These do not like water. And then you're supposed to know on this one, wait for it, there's something. Oh yeah, saturated. I'm going to draw a little, I'll draw it first. Remember how our little phospholipid had like one chain that was straight and one that was bent? Like saturated versus unsaturated. Okay, so you had your little carbon chain, and it's either saturated with hydrogens or not. Pretty simple. Lipids are simple. Just know that they hate water. So anything like waxes and stuff like that. Okay. And anything like waxes or oils, things that don't mix with water, that's a lipid. Okay. But our biggest one is phospholipids, and we'll talk more about phospholipids a little bit tomorrow because we're doing cell structures tomorrow.
Aren't the lipids on the inside of the cell wall?
Inside cell membrane.
Okay.
Yes. Yes. Well, yeah, the lipid portion is the inside of the cell membrane.
Is that when you're at the bottom? Is that like just the saturated side or does it not matter?
It's supposed to go what?
Like on a certain side, or you just go up there?
I just drew it like it's just an example of a lipid. That's a lipid. That's a saturated lipid.
What are subunits with the clock?
Oh, yeah. So that would be called a fatty acid. Oh, thank you, Chloe. I totally missed that. The monomer for a carbohydrate is a monosaccharide. Sorry. That's an A. Oh, my gosh. 2 C's. Monosaccharide. Saccharide means sugar. And then the monomer for this one is a fatty acid. Where am I going to put that? Although I don't agree that that's a true it's not really a true monomer. So it's kind of annoying. Because like our phospholipid, like it has a fatty acid chain in here and a fatty acid chain there, but see how they're not attached together like railroad cars or whatever? So to me, I'm like, that's not really truly a monomer.
Wait, so what are the subunits for the quaternary, whatever you say?
That would be like tertiary. Like tertiary would make up a subunit for that. So like when I'm finished folding my protein, if there's another related protein just like it and they bond together, like an alpha subunit and a beta subunit and a gamma subunit, then they make a big fat protein. Hemoglobin is a big one. All right, I'll try to behave myself tomorrow and not talk so much at the beginning of the hour so that we can have more time, more relaxed review session. Tomorrow is cell structures, which is organelles, just cell kind of processes, stuff like that, okay?
Bonding between the amino acids and the secondary